everyone. I, Ananya Verma, welcome you all to the talk on how to crack CSIR NET. Before we get started, I would like to request you all to kindly mute yourself. We, you will all have the opportunity to submit the text questions to the speakers by typing them into the message box in the Google Meet. You may send in your questions at any time during the talk. We'll collect these and address them during the Q&A session. It gives me immense pleasure to have the honor of welcoming this event speaker, Krithika Anbazhagan. She is a noted plant geneticist with over 8 years of experience in the field of crop research, who works as a crop physiology and modeling consultant at the International Crops Research Institute for the Semi-Arid Tropics, ICRISAT, India. Her research interests include enabling fit for purpose crop technologies and digital phenotyping tools for accelerating breeding programs and nutrition sensitive crops value chains. With over 18 publications, posters, and projects to her name, it goes without saying that she has contributed significantly to crop phenomics. Throughout the years, Dr. Krithika has also held various fellowships such as the Women Scientist Fellowship, the National Postdoctoral Fellowship from Department of Science and Technology (DST), as well as the prestigious PhD Fellowship from Council for Scientific and Industrial Research (CSIR). Uh, Ma'am, you may begin.
Hi, good evening, everybody. So I'm Krithika. Thank you very much, Ananya, for the introduction. So I'll probably leave the questions for later. Let me just start with sharing the screen. Yeah. So you can see my screen. Ananya, can you just confirm? Yeah. Okay. So good evening, everybody. It's nice that you are connected with Bioclues. It's a good opportunity to interact with people, also get to know what's happening in the field. So in this talk, I'm going to just briefly introduce you to how I had prepared or probable means which probably will help you in strategizing your preparation for the exam. Um, so my first question, I would want it to be a little more interactive, but maybe we leave it for later. So I would insist that everybody of you take some time and while you're doing the preparation for the exam, technically, you're reading the academic books, also have a look at why are you actually doing this or appearing for the exam and what is the motivation behind it. And because once you start PhD, it's a long-term commitment, and it is probably like chasing one question for, say, a period of 3 to 5 years. So to keep the momentum, you need more than just the fellowship or the good team or the reason to contribute to science. All of these have to align, but also along with it, you should be really motivated to do or driven to do this. So take some time and what do you call it? Dissect a little more and try to figure out what is the motivation and why you would like to pursue science or research, or if you're appearing for mathematics or physical science or any area for that matter. Yeah. And the next is to understand what is the purpose for this, whether you want to do it for lectureship or for research fellowship. And if it is for research fellowship, then what is the purpose of doing a PhD? If it is for job, then you need to appear or prepare for certain specific skills. If it is for being in academia, then you have a certain skills to hone. So take some time and think about these things as well while you prepare for the exam. So this is to say the why and what for the exam, and it's competitive. It's all India competitive. So the rigor has to be there, but to understand how you're going to appear and to keep the continuity in your preparation, I think we will talk about it in this session, about the textbooks that you have to access to, the knowledge sources or the databases that you can see and things like that. So other than the quality of preparation, you also have the quantum of subject because it's large now. So your sections will have your 10th standard maths, physics, chemistry, and then you will have much more detailed life sciences, which will have molecular biology, immunology, and biochemistry. So while the quantum is very large, you have to make sure that the quality is good. And this will come only when you thoroughly enjoy the subject. So even if you enjoy, say, immunology more than molecular biology, give it your best and thoroughly read it and use as much of imaginative skills as possible because that is what will help in tackling the multiple choice questions. When I gave the exam, which was like 2006-7, which is probably not relevant right now with the question papers format, but we had a subjective session for 3 years, which really made us answer the questions in a very, very concise way, which was not possible in our subjective papers regularly. So that meant that only if you know the subject, only then you will be able to tackle those questions, which is the same principle for a multiple choice also. Only if you know, you will know whether it is one of the four choices. So whatever you study and whatever you really enjoy studying, do it thoroughly. And some subjects you might struggle with. Maybe for me it was lipid metabolism. I really struggled through it. Like the structures, I would always get it wrong, or I would not know where the oxidation is happening. But yeah, so things that you know, do it well. Things that you don't know, still read it, but it need not be 100%. But go for it. Don't leave out certain subjects because it's hard. Just glance through. And if your preparation is rigorous, no, you will have a pattern of absorbing concepts. So trust your preparation and go for it. And one thing that actually helped me a lot throughout this phase, and it still helps me, is I just marvel at the science and what's going on inside. No? So when I was doing my master's, it was cell biology and immunology that kept me uh, very excited about the subjects. So molecular biology just changed the way that I was looking at a cell. And the more you know about how DNA polymerase works or how the quality check happens inside the cell, the more excited I was. But I struggled with biochemistry. But it so happens that I did my master's in biochemistry, which helped me pursue more. So now the nutrition studies that I'm doing now is more related to what I studied during my master's. But what gave me the push for CSIR was molecular biology. And I really wanted to do PhD at that time to pursue molecular biology and to see how a technology can be applied in real life. So when I joined ICRISAT, it was to look at genetic transformation of crops, and there I did a lot of cloning and all that, which was super fun. So the kind of joy that you get from studying science, I think if you enjoy it, it'll get very easier to study. And the more you gather knowledge, the more windows will open inside your brain, as well as avenues for career opportunities and things like that. And while most of them dread exams, CSIR NET is something that I thoroughly enjoyed. My master's exams I didn't enjoy as much, but this one has a kind of a mix, which gives you an edge. No, you can do a little bit of maths, you can do a little bit of physics, and then you have basic chemistry, then you have biology, and you have biochemistry, mol bio, immunology, all of these things. The cutting edge science as well as the basic science all comes together in this one paper. So it's a lot of fun. So take it as a fun. Don't feel beaten down by the pressure of it being an all India exam, or of it having 100 questions that you have to tackle 1 minute per two questions. I don't know what's the timing like, but don't get bogged down by it. Just enjoy it. Just enjoy the science, and I think it'll be okay. And don't get scared of failure. It's all right. These exams keep coming and going. And for doing PhD, you don't need the fellowship. It's a beautiful bonus if you have it. You're more independent. There's more regard. It's good for the CV and all of that. But if you don't make it, there's still a lot of opportunities. And if you prepare so well for your exams, then you can get a lot of opportunities outside. There are so many funded projects. So with so much of open connection over internet and Bioclues, you will know that there are so many opportunities open for pursuing PhD or for research assistantships. So don't get scared of failure, but success is fun. So good luck for your exams. I think the next speaker will have more technical stuff to give, and we can come back for more interactive sessions. Yeah, thank you. Let me stop sharing. Ananya, is it okay now? Yeah, yeah. Pranita can probably give more technical stuff. This was more like an overview of how to approach it. Yeah, thank you, thank you.
Ananya, have you started the recording? Yeah, can you hear me now? Yeah, yeah, yeah. I'm sharing my presentation. Can you see this? Yeah, okay. So, like Kritika said, um, uh, it's it's going to be uh, a conscious, thorough preparation. And most of us, see, there are exceptions, but I will say that most of us take two or three attempts to clear it. So be prepared for that thorough preparation. When I was preparing, I used to I used to study for like 24/7. Okay, so this exam is going to take that much from you. So now, how to start with it? Okay, all of you can see my screen, right? I'll be talking
Yes, ma'am.
about, yes, I'll be talking about the technical aspect of it, and I'm sure all of y'all must have gone through the syllabus. It is very vast, 13 units. I have only taken the headlines of it. Okay, the details you can get on net. So, from these 13 units, I will say please don't try to complete all 13 units. The first thing which you must do is select 8 units from these 13. Okay, I will second on this, like what Kritika said, ki don't leave anything and do everything. Do everything, but if you are selecting a topic, do everything in that topic. Don't try to mug up all 13 units. Okay, so first step will be to select 8 units, and there are some units, there are some portions which where the exams give a lot of emphasis. These are the first 4 5 units. Okay, so I will suggest not to leave biomolecules, not to leave biochemistry, not to leave molecular biology, and a little bit of signaling also. They ask many questions from cell signaling and cell communication. So these are some units which I will suggest, if you can do, please don't leave these units. Now, If you are a botany person, of course, you will take up systems physiology plant. And if you are a zoology person, you will take physiology animal. Okay, systems physiology. So you can select any one. If superficial questions are there, you can attempt them, but please leave one of them. If you have taken both the subjects in your graduation, then it's okay. But like in our case, we had to select only one. So I was a botany person, so I did not touch animal physiology at all. So I will suggest something of that sort. Then genetics is there. You can uh, either do it or leave it. And you, according to your expertise, your liking, you can select the units. Okay, now about unit 12 and 13, I will talk about that later. So just quickly we'll see what is the pattern. There are three parts to the paper: part A, part B, and part C. Part A has 20 questions related to general science, mathematics, reasoning, and all. Okay, so I'll talk about how to prepare for this part. So you have to attempt 15 questions from there. Then the second part has 50 questions and you have to attempt 35 questions. Part A and part B, each question is 2 marks. Okay, so 30 plus 70, so 100 marks over here. Coming to part C, it gives you a lot of choice. Out of 75, you have to attempt only 25 questions, but it is 4 marks for each question. So again, 100. So you know all this. Now, how to prepare for part A? You have to do only 15 questions. And there are very wonderful YouTube videos on this part A questions. The maths of class 9, 9 to 11, 9 to 10th, 8th, even 8th mathematics is there. So just revise all your mathematics. Just do small portions every day. One or two videos you see. And when you are doing this, make a register and write all the formulas. See, you can't remember, and at the end, before the exam, you will not get time to revise all the topics because the syllabus is vast. Okay, so make a register for your last minute revision and write minimum over there. Don't write too much over there. Only formulas. Just put the topic, formula, topic, formula. There are questions based on speed, distance, and time, trains moving. Uh, then there are questions on angles, like clock में इतना बजा है तो इसके एंगल कितने होंगे, आर एंड मिनट हैंड के बीच में कैसा एंगल बनेगा. These are very complicated questions. You can't do it, but if you know the formula, you will do it in minutes. Okay, so there are all sorts of questions. So just give, like, when you are sleeping, when you are free, just we are browsing in our mobiles, just watch one or two videos every day, and if there is a formula, note down in your register and then forget about it. Okay, just understand it and then forget about it. We will take this register in the end, before going for the exam, 2 days before the exam, we will do that. Okay, so note the formulas and do that. For part C has many analytical questions, questions based on techniques. They will give you some results of experiments and they will ask you to interpret it. Basically, these kind of questions are preparing you for uh, your research. Okay, that if you are planning an experiment, you are getting a result, it's okay, but you should know how to analyze the result. Okay, if you're getting a band, certain band in the electrophoresis, um, what does it mean? So some questions of that sort are there. So for that, if you go through science news, sometimes just the abstract, just, and what are the techniques that they are using. Okay, and understand the principles of that technique. Don't go into too much detail, just the principle. Like for example, if we are talking about this gel filtration, so you just have to know the principle that the smallest molecule will come out first. So this is the principle you should know. And in part C, there are also some match the columns questions. which are very, sometimes they are very easy, sometimes they are tricky, but sometimes you can solve them. Like if you know two of them or three of them, you can solve it if you don't know the fourth one. Or yeah. So just try. Actually they are making the options very intelligently, but sometimes if you are lucky, if you know the two of them, you can get the answer. So that is one thing. Now when you get the paper in your hand, what you have to do, okay, be very calm, don't worry, only attempt the portions that you have studied for. Okay. So the sequence will be which part, first of all, you should always go for part C. Why? Because each question is giving you 4 marks over there. The questions will are analytical types, so you will need your brain. So fresh, your brain is very fresh, you are sitting for the exam, first go for part C. Okay. Very big questions are there. Sometimes you have to read very calmly, read the questions. If you are not understanding a question after reading one or two lines, then you leave it. Save your time. Don't read the question fully if you're not understanding the first. See, when we start reading a question, we kind of get we understand it, that will we be able to solve it or not. So you will get that instinct yourself. So if you feel you are not understanding anything from that question, just leave it, don't even read the question. Okay, save your time over there. Okay, then match the column. I told you, you can do it that way. uh, and see, the questions are really very sometimes they are very easy and sometimes very tricky. I will discuss two questions with you over here. There was this half page question. The story was written that they were discovering how DNA replicates and all. It was a half page question. Normally you would be afraid of reading the question only, but you know what the story was? The story was the full Michelson and Stoll experiment. If you have read this DNA replication Michelson and Stoll experiment about semi conservative model. So they had explained that, and the options were, like, the question was, now what you will interpret from these results, and the option was DNA replication is semi conservative, DNA replication is conservative, and whatever. So sometimes if you are lucky, you can get such easy questions over there also. So I first thought, let's leave this question because it is a very big question. But when I read it, I was understanding, and I was feeling that it is an easy question. I read the whole question, and then I couldn't believe myself that they are asking this. So sometimes even the questions, such easy questions, are also there, and it is giving you 4 marks. So just be very intelligent how to select your questions that you are attempting. Then there was one question about Krebs cycle. So I don't know. I am studying Krebs cycle from class 11th, and every time I know the whole everything about Krebs cycle. किसके बाद कौन सा मेटाबोलाइट आता है, कहाँ कौन सा एंजाइम है, कहाँ कितने एनएडी निकल रहे, कहाँ कितना एफएडी निकल रहा है, सारा रटा हुआ है, everything. But I couldn't solve that question. They had given the structures of the metabolites, citric acid, oxalic acid, and all. So they had asked which is this, the order. They had asked, they had given the structures, and they had asked the order of the metabolites. And so I thought, okay, I don't know the structures exactly, but I know that ketoglutaric is 5 carbon, oxaloacetic acid is 4 carbon, citric acid is 6 carbon. So I thought I will do it that way. But they tricked us. They gave two options in which the position of citric acid and isocitric acid was interchanged, and exactly I did not knew the structure of that, and I couldn't solve that question. So I'm just I just want you to know that that specific, that is the level of specificity that they are making their questions. So even now it is not subjective, it is objective, but again I will say that they questions they are making very intelligently. And so please, whatever topic you are studying, do that thoroughly. Okay. So yeah, so I told you about last minute. What you must do, keep 2 to 3 days, last 2 to 3 days you close all your books, and just don't, even if you're traveling somewhere abhi to kya, in your state only, in your district only, you can get the center. But we used to go to somewhere Bhopal or Nagpur, we used to go. And when I was traveling, I didn't carry any of my books, only that one register or some pages where I had written important points, important notes, formulas. For keep 3 days for those formulas, revise those formulas. Also important points like some principles, something, anything you feel that you will forget in the end, some structures or something that you can revise in the last minute. CSIR net usually doesn't ask you about the scientists, that who discovered this, who discovered that, so you can leave that. All that you can leave. And if you're preparing for CSIR net, I'm sure you will clear DBT, you will clear ASRB net, because I think CSIR, the standard of CSIR net is the highest. It covers everything. So if you're preparing for that, you will be able to clear any other JRF exam. So that's all I want to talk to you. And about the books, so I had my favorite book was Leninger. The first, second, third, fourth, all, mostly all the first four units you can cover from Leninger, full Leninger. And the questions that are given at the end of the chapter, try to solve them. Then they have given some information in boxes also. So that is also very important. I have noted many times they have asked questions from those boxes. So go through don't leave those boxes, just read them also. And what carp also is a good book for cell biology. Leninger for all biomolecules, molecular biology. For developmental biology, Gilbert is a very nice book if you want to do that. uh, yeah, so these are the books. For immunology, Cube is there. So that's it from my side. Any questions, they are welcome.
गुड इवनिंग मैम।
Yeah, tell me.
मैं मंडला से बोल रहा हूँ।
जी।
और लेलिंजर क्यूबे में मैंने जो बुक मंगाई थी उसका प्राइस तो बहुत ज्यादा है, तो आप बता सकते हैं मैम समराइज में किस तरीके से
No, don't buy these books, please don't buy these books. If you can join some library, you can issue your the Leninger from there. Otherwise you can get the soft copy, take a print. That is the best thing. uh, Take a print, it's worth. Soft copy of Leninger is available. Yeah, yeah, it's available. If you want, I can share. Leninger को रामायण की तरह रट लो. Nobody will stop you from clearing net. That's my motto. Yeah.
Hello, ma'am. Ma'am, Leninger book is so much hard to understand. So please recommend some other easy book.
Yes, actually, it is not hard, beta. It has lot of information. And if you can't read, then see, it has very good diagrams. Go through those diagrams. Uske
Yes.
niche jo likha hai, go through that. It has explained it very nicely. It is, I will not say it is hard. It has lot of information. So once you read and just mark, what portions you have to read when you are reading it again, what portions you have to leave. There is some extra information there also. So other book, I don't know. I have studied Leninger, so I will. And Pathfinder, about Pathfinder, it has given things in very concise way. Like For me, it's easy for Pathfinder. But if you go with Pathfinder, I think it has missed the details. So for the details, I will suggest these good books only. Gerald Karp is there. And yeah, I don't know if Kritika can suggest some other books.
I think मतलब मेंटली तो बहुत सारे चैप्टर्स
I can't hear you, Abhijit. Abhijit, what you are saying, I can't hear.
हैं एलिमिनेटर इतनी बड़ी बुक में बहुत सारे चैप्टर्स ऊपर में एक साल हो जाएगा पहले चीजें भी
Maybe you can type the question, yeah, Abhijit.
Abhijit, yeah, type the question. I'm not able to hear. Uh, Prash can
Hello,
say, yeah.
Yeah, I'm sorry,
hello.
Pranita. Thank you so much, Pranita and Krithika, for excellent inputs. This is for some of the folks who have, message saying कि that pdfdrive.com से download कर सकते हो and all those kind of things. Please, please don't do this. This is all, infringing copyrights. Please understand this. As Pranita has rightly pointed out, she can offer you, she can suggest you that she has a soft copy. She can give it to you by email, in person, but not in public group. Okay, so
Yeah.
please learn the etiquette and ethics of open access, and as well as copyright issues. Please don't download from any of these particular resources unless otherwise required. Even unless otherwise required भी नहीं चलेगा. Okay, we at Bioclues also ensure that we teach this kind of etiquette and ethics to everyone. Okay, especially Ranjit Chauresia, please please remove that particular link. Please don't encourage anyone to download any of these particular free books, which means you are absolutely infringing copyrights. Even if someone files a case, you'll be put up behind bars. Please understand this. These are all very sensitive issues, which many of you don't know. Okay, sorry
Yeah.
to take this talk otherwise. Thank you so much.
Yeah. Yeah. Yeah.
Thank
Yeah,
you.
in continuation what Prash sir is saying, see, there is a Government of India website called EPG Pathshala where all the notes are provided, all the topics, topic wise. It's like, every student can access it, freely. It's provided by the Government of India for all the PG students. That's the best source you can start off with to start preparing for this exam. Okay, you don't have to look for, buying books also. So when you have access, you still want to go with something, all these websites where you have a problem with copyright. See, discussing among your peer group is fine, but on a professional platform, I would advise not to talk about these things. But still, you have certain websites called EPG Pathshala by the Government of India, because I'm repeating this, I've been repeating this to my students also. So please access them, and at least for those who don't have any books, or are in a remote village, or remote part of the state, at least you have this, you have phone with you, right? You have already, everybody has access to these phones, or at least those who have it, just take it, download, and then take a print. Okay, fine, thank you.
Hello.
Anyone else, any
Uh,
other question? Yeah, please.
yes, yes. Good evening, ma'am. Good evening, everyone. I have one question about the how, ma'am, how to manage time and how much time it will require for in a day. Because I recently I got an admission in PhD, and now I'm busy in my research work, but I'm not clear till now that the CSIR exam. So, and it is too difficult to me. A whole day I'm working in a lab, and when I come back in my room, so it is not means not properly prepared yourself to read something, because I feel so tired and just fall down and sleep. So it is too difficult to me now. Please suggest me something.
Pranita, will you take it? I'll
Yeah, yeah. I'll talk about it. So, if you, it will take a little bit more time. Like if two or three attempts more, it will take. I will take. You make a timetable, uh, and don't wait for the evening when you are in your lab. You have some time. If you have some time free, first of all, you make a timetable. How, what units you will be covering, how much time. So that will give you an idea that if you are not able to complete a topic in a particular time, So you will know that you are lagging this much behind. Now you have to cover it up. In the morning, complete some topics. So, and yes, you have to take a little extra mehnat karni padegi. I know, I understand. Doing it together with PhD is difficult. When I was preparing, I was working and I had a child, so it took me 4 attempts to clear it. So I think it will take more number of attempts. It is happening 2 times a year, so it's good. You have to make a timetable and see, you have to think, you have to, like 24/7, you may not be studying, but you may be planning, you may be thinking कि आज ये कर दिया है तो यहाँ का टाइम चला गया, तो you must make compensate for that time, and it will require a little bit of hard work, but it will happen. If you want to do it, it will happen.
Yeah. Anuj, can I ask what is your research on?
Yes, ma'am. It is my research is on, and I'm working on promoter characterization now in wheat.
So, so it's bacterial cloning and stuff.
Yes, yes, bacterial cloning and some sequencing bioinformatic work also.
Yeah. So, I think in the day when you're working, there will be already certain questions that will pop up to your head, no? So, but then
Yes,
go back
yes,
and
ma'am.
read about them. Don't skip questions that cross your mind, even if it is related to the work that you're doing. And when you're flipping through the book, no, like if you have Watson handy, and if you're flipping through the book, then you will read other things that will, you will subconsciously absorb it also, and interest will keep you engaged, so your mind will be engaged on these kind of questions that you'll be curious about. Talk to your colleagues, talk to your friends, talk to juniors, seniors, discuss a lot while having chai. So be on that momentum, don't lose the momentum. If we shift gears and we are talking about something else entirely, then it takes effort to fall back in line. So keep talking about the subject, enjoy the work that you're doing. It will keep adding on to your knowledge base, no? So yeah, it's hard
Yes,
work,
ma'am.
but then it will come through.
Yeah. Thank you so much, ma'am. Yes.
We have a question in the chat box by Rohan. Uh, how to use the old question papers. Could you please, one of
Yeah,
you,
I
take the
have
call?
never used old question paper, yeah, so I don't know. But I've heard from my friends that it's very useful. It gives an insight on what is recurring. And those are like short questions that you can go for. So I think it's like your plan B. Although plan A is first hand preparation, plan B is to know that these things will work.
या ओल्ड क्वेश्चन, डोंट थिंक दैट ओल्ड क्वेश्चन हम इसलिए पढ़ रहे हैं कि वही क्वेश्चन रिपीट होगा. Old question papers will help you to understand how you are preparing. Is your preparation up to the mark? Because when I started preparing, I took a paper and I was all lost that I can't clear this exam, because the questions that they are asking, they are very specific, very basic. So that will lead you to the direction how to, like what areas you should how to prepare. हम पढ़ लेते हैं, जैसे मैंने बताया आपको, क्रेब साइकिल हम पढ़ते आ रहे हैं हमेशा से, लेकिन उसमें वो ऐसी चीज पूछ रहे हैं जो हम ध्यान नहीं देते हैं, है ना. सिग्नलिंग, जैसे सिग्नलिंग मैं बहुत अच्छे से पढ़ती थी, बट सिग्नलिंग दे आर आस्किंग इन वेरी मच डिटेल, बहुत सारे सिग्नलिंग पाथवेज वो कॉन्सेंट्रेट कर रहे हैं. तो वो आपको ओल्ड पेपर्स विल टेल यू कि ये भी पढ़ना है, इतना डिटेल भी पढ़ना है. ओल्ड क्वेश्चन पेपर्स का ये मतलब नहीं है कि रिपीट होगा क्वेश्चन, एंड इट विल ऑल्सो गिव यू एन आइडिया कि कौन से एरियाज पे कई बार वो ज्यादा फोकस कर रहे हैं. सो दैट वे ओल्ड पेपर्स विल हेल्प.
And another point I'd like to make is that old papers will let you know how the questions are framed, basically. Like ये बहुत सारे applicative होते हैं, direct questions बहुत कम होते हैं, only 60% direct questions होते हैं. बाकी के जो 40% which you have to compete for to get a very good score is applicative question. मतलब A to B का C का, या भी script cycle के example ही ले रहे हैं. So if you come with A, B and C, they'll ask question on C rather than A and B. It's like more applicative. So you have to, old question papers will help you know, okay, how the questions are framed. That is what the all question papers will help you. You have raised your hand. Please ask your question. You can unmute. Kushal, uh, unmute yourself and please ask.
He left the meeting only. One more
Yeah.
point I missed, I had thought of it, that there is negative marking. Huh? All of us know that there is negative marking. So, like, if you just look into the cutoff, it is above 55. I don't know recently what is the cutoff, but yeah, my during my time it was 53. So I just made a line that 60% of the paper, if in one part I am able to do 60%, then I will not push myself. Like, first we will do all the questions that we know that they are correct. Then you just calculate and see if it is okay. Then don't push yourself for a question that you are not sure of. So also that will sometimes help. I used to do like 55 to 60% अगर हो गया है, जैसे फर्स्ट पार्ट पे 15 क्वेश्चंस करने हैं, if I'm able to solve 9 or 10, then I will not push myself for the 11th question if I'm not sure, because it will cut my marks. And especially in section C, because there 25% means 1 mark. So that also, keep that in mind.
Indrani, would you like to please say something?
Yeah, sorry for my voice. I hope I'm audible, ma'am.
Yeah.
Yes.
Okay, thank you. Just as our speakers have said elaborately of all the preparation types and everything, I just want to give a small wrap up. In the recent trains, like my juniors and I have also tried past 1 to 1 year. So now the questions that are being decided, it is being shifted to National Testing Agency. So they are putting more stress now on statistics. You see,
Mhm.
from any life science, any field, if you say statistics plays a huge role there. We are not aware of that application. They are putting those kind of questions in section B and section C. Based on statistics, they are putting the questions. Even if you see from the ecology side, we have been seeing the trend. Like ecology is like a major question. Like most of the questions come from ecology, and ODM is a very good book for that. But once you study with all these books that our speakers have already mentioned, you make sure you have 6 to 8 months thoroughly to go in depth with with all the concepts. Say, for example, cell communication and signaling, as Pranita ma'am was saying about that. What all the components are involved in that particular signaling pathway? All what all techniques can you use for detection of the particular protein molecule? Everything in details with application in every point. Okay, make sure this kind of things they are looking more. There are no more straight questions. There are no more even the paragraph based questions in C parts. They are changing it with, they're connecting like 2 3 concepts together and then putting up the question. So make sure you are well aware with all of the concepts that are behind in each of these chapters. Okay, so that's all I just wanted to add up. Yeah, thank you for giving me this opportunity and wonderful speakers we had, ma'am. It was surely a wonderful session we had, all of us. Thank you, ma'am, for giving me this opportunity.
Any other questions by the participants?
Um,
He asks about percentage and percentile. Uh, Rohan, can you ask what is? I mean, are you asking a statistical question or is it something in the exam?
Actually, this year they have not mentioned the cutoff percentage of CSIR. They have given it in percentile.
Acha,
Yeah,
okay.
ma'am.
I think they've started it from
Yes,
this
ma'am.
year only, this time
Yes,
only.
ma'am.
So that
Actually,
I
they have
think he's
started.
asking.
Yeah, ma'am. You're correct. They have started in the year of 2020. After COVID, they have changed that plan.
Okay.
And this is only for life science. For others, they are giving percentage only.
Oh, is it so, ma'am? Then, oh, only
Yes,
for
yes.
life science they changed it.
I saw, yeah, for others they are giving percentage. I did not see
Percentage.
the cutoff in 2020. Only this time some of my friends had given. So I saw. And only life science they are giving it in percentile.
Yeah, Rohan says 94 percentile life science cutoff this year. I don't know what it means. Yeah. Do you have an idea? Prashant, do you know about this? I have
No,
no.
not really. Actually, not really, Kritika and Pranita. Basically, what the question pertains is more towards cutoff parameters. So percentage is more mathematical. So here, since in CSIR net you have more number of questions with kind of negative cutoff marks,
Hmm.
percentile is more appropriate. That was what the new educational policy, which was framed in
Hmm.
2019.
Huh.
That was what it was framed. So ever since, all the marks are no longer put up in percentage but in terms of percentile. So percentile is less than what we call it as a percentage. So which means if there are 180 questions and if the person gets say 90%, then the
Hmm.
percentile would be somewhere around 86 percentile. Okay, so
Okay.
85 or 86 percentile. So percentile is more appropriate. So absolutely nothing to worry. So hereafter, across all the graduate programs or all these competitive exams, you happen to get to see only the percentile, not the percentage. So in a nutshell, percentage is more relating to the numbers, and that's where you give it percent. Per is
Yeah, per 100.
by 100.
Yeah.
Percentile is nothing but less than 100
Proportion.
actually. That's where your the number of questions are appropriate. So this is as per the new educational policy. That should not be a kind of a worrisome factor. In your mind you can keep your percentage issues, but what you get in marks is percentile. Yeah.
Yeah.
And I just have one quick comment if there are no questions. Actually, while thanking these revered speakers for the wonderful inputs, there's something called positivity. That you should always go through your preparation process. And please understand that some of you might be preparing only for lecturership, or some of you might be preparing for NET, national eligibility test, and all those kind of things. Please understand that post-COVID times, there has been some kind of behavioral problems. As soon as you aim for PhD is not just a kind of a philosophy or doctorate. You should always look into 6 important traits that we often say. Okay, one is patience, honesty, discipline, perseverance, humility, and dedication. Okay, so if these 6 traits should always keep it in your mind, and that's where you can probably look into PhD. From our survey, those people who have cleared CSIR net at the helm of third or fourth year of their PhDs,
Hmm.
whilst submitting their particular thesis, there were many cases, many students who didn't who don't even submit their thesis because of some vivid reasons as well. That's because maybe these kind of 6 traits backed up by positivity is really missing in them. Okay, so please ensure that I know you might be preparing for so-called CSR net, but you should always stay positive no matter what kind of things uh, you might make up. And for these 6 traits of PhD, you could always start your day pretty bright. Always maybe you could always have a meditation during 4 to 5 in the morning. That would probably for 10-15 minutes of meditation, then start your day. And as Pranita says, 24 by 7 you keep discussing science, keep asking, certain questions that are unasked as well. That would probably allow you to reach, pinnacle of success. That's it from my side. Thank you.
Maybe I would like to just add very quickly one small point.
Yes, please.
Yeah. So we were discussing about the negative marking, but if there are like you have 5 such questions where you are not absolutely sure of one perfect right answer, but you are confused between just 2 out of 4 options, then it would be good to go by your instinct for such questions where you have ruled out 2 other wrong answers. And maybe you can, in that case, you would have a higher chance of basically scoring at least half of your answers correct. And in that case, even if other half are wrong, you would only have 25% negative marking generally, or 33%. So, but if you are going by your instinct and have understood the question correctly and ruled out 2 other wrong answers, it would be good to attempt at least some of such questions where your instinct is suggesting you the correct answer. And that can be subjective actually, but you can think of that option also in some instances. Thank you.
Yeah, you need a very cool head when you're going for the exam. I mean, anxiety will not help.
Yes, sure.
And yeah, so it's just an exam. Take it, prepare well, and go for it with a cool head. Yeah, only then you will be able to sense your instinct also.
Yeah.
Otherwise,
Yes.
it's
Yes,
in
sure.
anxiety you will.
Right.
Yeah.
Yeah. Yeah. Thank you.
Yeah, thank you, Pushpinder. And as for the questions right now, someone asked about what are the uh, specifics of the declaration. It will be on the criteria for the declaration will be on the website. Please just read that. Okay, reading is what is more important before you ask such questions. And as for Swati, please, this is not relevant for the webinar right now. You can have a discussion later. Okay. And Ananya, please take over.